Okay, perfect. Well, I'll get rolling then. So again, I'm Dave Stark. I run the agriculture business of a company called Holganix. My background is I have a doctorate in molecular biology of all things. I've spent now 41 years in ag and ag technology. I don't have my camera on because I'm just too old to be seen. Not really. I'm in a hotel and I don't want to over stress the free Wi-Fi because that's all that's available. So I want to talk about microbials and really more educational, how putting life back in the soil is so important. And I know there's a lot of things out there in the whole field of biologicals and microbials. A lot of them are the same. A lot of them are lacking, but there is truth behind it. And that's really what I want to go over in terms of what to look for. So this is a chart. And by the way, at the bottom, you're going to see on a lot of my slides, the little print that will have someone's name and in a journal and such. What I'm showing, because I am a scientist by training, even though I have run businesses most of my career, I go to the scientific literature. So what I'm showing you is not my science. It's not my company's science. It's generally the peer-reviewed, published science that's out there that's generally done by universities. And I encourage you all to read it if you have trouble going to sleep at night because it's pretty dry, but it's really where we find the information and the truth. So this is a chart of the different types of microbes in soil, starting from the smallest, the most abundant, the fastest to grow bacteria, down to actinomycetes, which are kind of bacteria-like, to fungi, protozoa, nematodes. I mean, there's others, but these are the main types. And as you go down the list, they're larger, they're slower to grow, they're less abundant. If we look at it, the number of these things in a gram of soil, so a gram, if you know the candy M&Ms, you know the chocolate with the candy coating, one M&M, you know, not one with a peanut or almond in it, but just the chocolate, that's about a gram. So in a soil that size, if it's healthy soil, it can have over 1 billion bacteria. And you go down the list, you can see. So if we look at the biomass over an acre, and yeah, you can tell I'm south of the border, so I do pounds per acre. So forgive me for that. But this is in the top 6 inches per acre. Look at the range. So bacteria can be over 2 tons. Fungi can be almost 7 tons or 9 pounds. That's huge. And this really is indicative of the biological health of the soil. So what's going on? Well, there are things that happen that change the microbes in the soil all the time. They're constantly in flux. Some of the things we do, some of the things we have no control over. So for example, when we till, and we're going to talk about this, disturbing the soil changes the microbes in the soil. Our use of chemicals and fertilizers, so fumigants, of course, sterilizes the soil. Overuse of fertilizers. I'm not bashing fertilizer. They're a blessing. We need it. But overuse really changes the biology. So there are things we do to make things worse, but there are things that just happen. We have no control over. Winter. Microbes are full of water. When water freezes, what happens? It expands. So it kills a lot of the microbes, but not evenly. Some microbe types and species tolerate freezing better than others. Another thing that happens is flooding. The microbes you want in your soil need oxygen. 48 hours underwater, the oxygen is depleted, so they start to die. And you can tell when the water drains and you smell it before you see it, like the bottom of a swamp, that tells you that ground has gone anaerobic. Basically, it's lost its oxygen. It's dead of the things that you want. The things that can grow without oxygen, unfortunately, tend to be the pathogens. So we don't want that. We want to bring the life back. And then just to make this more complicated, plants 6 to 10% of the community in the root zone of microbes is different plant to plant. And I don't mean corn, to wheat to soybean to tomatoes or oak trees. I mean corn hybrid to corn hybrid, soybean variety to soybean variety. Plants actively recruit and repel the microbes they want in their root zone. So, look, I usually do this. I'm not a salesperson, but I want to tell you, this is what we do. So I guess I ought to show you. We make a product that has everything I showed you in that table. Really, over 800 species. It's really a couple thousand species: bacteria, actinomycetes, fungi, protists, beneficial nematodes. We DNA fingerprint each batch, so we know everything in it. These are living microbes. They're not dormant. They're not spores. We put food in there: molasses, amino acids, micronutrients, some humic and fulvic acid. And just to make it even weird, because these are living, after we make them, we have to refrigerate them. It's the only way we can suspend them. This is a clue. The next time someone's coming up to sell you something that they say will do everything that you need in the soil, is it a refrigerated liquid? Because if it's a powder, or if it's in a jug or a tote that you can keep tightly sealed at room temperature, it cannot have all of that biology because they need oxygen. So this is what our product does. So enough of the commercial. This slide shows some of the things the biology does in the soil. It's the microbes that makes the soil function. Just like the microbes in and on us are such an important part of our immune response, of our defense. It's not everything, but it's a big part. It's the microbes that make the soil work. We can't talk about all of these, but we're going to start with nutrient efficiency. And why? Because it stinks. Nutrient use efficiency. So you've heard the saying, less than half the fertilizer you put on the ground actually goes to the crop the year you put it down. That's true, and it's generous. So there's different published numbers. The number here for nitrogen, I think, is high. It's more 20 to 30%. Phosphorus gets bound up quickly in the soil. Same with potassium. Now, these are averages. This doesn't mean this is what happens on your ground, but these are averages. And if we think about, now this is again the US in 2021, about a $20 billion fertilizer market. That means over $10 billion of really our farmers' money not doing its job. So how do we do better? There's a lot on this slide because a lot went into it. This is the summary of 230 peer-reviewed, published scientific studies looking at how nitrogen is fed and used by corn, rice, and small grains. This is a huge body of evidence. So if we start with what's here in the black box, in the middle, where we don't have good biological activity in the soil, we put nitrogen on the ground, the plant takes some of it up, an awful lot of it is lost. Now, it may still be out there, it may be tied up, but a lot of it is lost in erosion. With rainwater, it volatilizes. It's not efficient. The engine isn't working. But where we have biologically active soils, go to the right, we see the loss is much, much less because the microbes are stabilizing the nitrogen, keeping it in the root zone and feeding it back. The bottom line is there in the green box. Biologically healthy soils produce greater corn yields per unit of fertilizer input. So what's going on? Let's take a closer look at what this SON is, soil organic nitrogen turnover. Why are microbes doing this? Come on, go to the next slide. There we go. I like pictures. So there's a lot on here, but again, you see the picture. So we start with fertilizer. They're in the lower left-hand corner. It's usually some form of ammonium or nitrate or both. You put it on the ground. The first thing that happens, remember the bacteria and actinomycetes? Those are pictures. That kind of orange one is a close-up of actinomycetes, and there's bacteria. They come in different shapes and sizes. They're very numerous. They're always hungry. They take the first bite of anything you put out there. So when you put fertilizer on the ground, if you have a biologically healthy soil, they eat it. They eat more of it than your plant does, and that's a good thing because the microbes live within half an inch of a living root, and they immobilize it and they stabilize it. And so they convert it from ammonia and nitrate into organic nitrogen, which is protein. Now, it's a good thing because they live in the root zone. It's a bad thing because we're going to talk about carbon to nitrogen ratios. Bacteria and actinomycetes, on average, for every nitrogen, they have 5 carbons. As a biologist, I can tell you they're nitrogen pigs. They love it, they tie it up, they bind it. They immobilize it, but they don't let go. Now, the things that eat bacteria, though, that's different. So if you go to the right across the top, all that white feathery material, that's fungi. Now, I'm not talking about yeast, which is also a fungus. That's single cell. They're important in the soil, but they don't do this. They don't go through the soil, degrading organic material, etc. When they eat bacteria, their carbon to nitrogen ratio is 3 times less. So 1 nitrogen for 15 carbons. Now, the real predators are there at bottom middle, the protozoa and amoeba. A single protist can eat 10,000 bacteria a day. So they love bacteria, but their carbon to nitrogen ratio is 6 times lower. So it's 1 to 30. So they're eating all this nitrogen-rich bacteria. It's actually toxic to them. They have to get rid of it. It's actually just like us. Not to be indelicate, but urea and urine sound similar because that's us also getting rid of nitrogen. We're also 30 to 1, like the protists. So they eat the bacteria that have immobilized the nitrogen, they have to get rid of it, and they spit it out as ammonia and nitrate. What are the 2 forms your plant root takes up the most readily? Oh, ammonia and nitrate. Where does it happen? Well, the bacteria live within half an inch of a root, so in the root zone. You know, like I like to say, God designed this perfectly. This is the engine that God designed. We want this engine to function because this is what gives the best nutrient use efficiency. Now, that's not all. Again, I keep talking about God's design and not millions of years of evolution. That's a whole nother conversation. Molecular biologists have a lot of expertise in that area, and I have studied this and I have changed my tune. But again, that's something we can talk about offline. Here's another thing that just blows my mind. So this is work that's out of Rutgers University from a few years ago. By the way, I removed about half of the graphics from this slide to simplify it, believe it or not. It's real simple though. So what happens? Microbes, and when I say microbes, I mean bacteria, actinomycetes, fungi, all of them, and the plant root are constantly sending out and receiving biochemical signals. So they're chemical signals. When the signals to the plant and the microbes are, hey, the microbial population is now at a certain level and it's diverse. It's not just a few species. It's not just bacteria, not just fungi, it's diverse. That's called quorum sensing. Once quorum sensing is hit, things change in the soil. And I'm going to show you pictures of this. One of the changes is the root tip starts absorbing microbes. So yeah, there's a whole farm underneath the ground out in your field. The plants are farming the microbes because the microbes mine the soil and they eat them. They're eating the microbes. They absorb them, and they're at the tip of the root. They strip off the outer coating called the cell wall, which is very nitrogen rich, very rich in protein. Now, some of the microbes just die. Actually, a lot of them. They just explode and they release all their contents. This is also good because every nutrient your plant needs, N, P, K, S, all the micros, so do the microbes. So now the plant's not getting just nitrogen, it's getting everything. Some, though, survive. They travel up the root, they go to the bud where the root hair is going to form, They secrete a hormone, which then induces the elongation of the root hair. They're spit back out in the tip in the soil. They remine the soil for nutrients and cycle back again. So there's a couple different ways microbes help with nutrient use efficiency and feed the plant, and we can see it. So these are pictures that I've got. So, in your beans and legumes, you get the root nodules because they help fix nitrogen. And for years, guys were telling me, Dave, I see so much more nodulation when I use your product. And I just thought we were dumb lucky. Well, we are dumb lucky. But what was really happening is our product, because it's diverse, is a quorum. We trigger quorum sensing, and the plant root has to take up the rhizobia and the other bacteria to form the nodule. So we're triggering that quorum sensing and absorption faster. And that one picture is actually from a customer in Ontario, Canada. But we can look even more. So this is a picture. This came from Iowa. These are corn roots. And you can see the ones in the center and to the right. It's too blurry to zoom in, but there's almost no root hairs. And there's almost no dirt stuck to the roots. But where we used our product, the three plants on the left, there are lots of root hairs and that dirt glued to the root. So what this tells us, by the way, I like this picture and the one before because you didn't have to send samples off and spend a lot of money. All you needed was a shovel. So what this tells me is this dirt does not have biological activity diverse enough to really drive quorum sensing. We're not getting root hairs. Another thing quorum sensing does is the plants and the roots exudates and the microbes make a glue that glues the soil together, and that's called the rhizosheath that you see there on the left. That's the action point in the soil where nutrients are exchanged, the plant's protected from drought. So again, you can see how biologically active the soil is often just by using a shovel. Now, I did this for nitrogen. Same thing, phosphorus, potassium. There's generally a lot in the soil. It's not available, as many of you know. It's in a form that the plants cannot access. Microbes can. Microbes mine the soil. Plants farm microbes. They work together to feed each other. Now, so we go out and we do a lot of soil sampling. And here's just an example that's looking at the different nutrients with and without our product. And you can just see the availability of NPK, et cetera, organic matter. It's all improving through use because the microbes are doing their job. And that's what we want. Microbes, when you have this, improve fertilizer use efficiency. So they're a tool to help a producer start backing off their NP and K inputs. There is no formula because your ground is different. And what you just heard, from before this talk, Zach showed, even the ground in your field is different. So it's a tool that you can use to slowly back down and pencil in where the inputs and the yield bump maximize your profitability. And here's an example again from third-party data in Iowa, but I've got a lot of data like this. So I'm going to move on because microbes do more than just release nutrients in the soil. They also release it from all your crop residue. You have a lot of nutrients not only locked in your ground that we just talked about, but in the debris that you've got sitting on top of the ground. So for example, every 40 bushels of corn produces a ton of stover. In a ton of stover, 17 pounds of nitrogen, 4 pounds of phosphorus, 34 pounds of potassium. Potassium is a very important mineral for stalk and stem strength, and some sulfur. So if you have 200 bushel an acre corn, you've got 5 tons. You've got a lot of nutrients that you've paid money for that you want to return to the ground for next year's crop. Not one speck, one atom ever gets released unless a microbe breaks that residue down to the basic chemical forms that the plant root can take up. So now we're back to the carbon-nitrogen ratio that we talked about earlier. So remember, bacteria, about 1 nitrogen for 5 carbons. There are a lot of residue degrading microbial products out there that only contain bacteria. And it drives me nuts, candidly, because bacteria, first of all, don't have the right biochemistry, not compared to fungi, to efficiently break down the polymers that make that residue so hard to degrade, cellulose and lignin, but fungi do. Plus, you're going to put bacteria on corn stover that has 1 nitrogen for 55 to 60 carbons, or your wheat straw, which is 1 to 80. Where do they get the nitrogen? Well, they either are not doing the job and they're not doing it well because they don't have the biochemistry, or they steal it from your ground, which is why you can get yellow corn. So what we need are fungi. And that's something that we also formulate. It's a very fungal rich product. You can see on the right just the difference. You want to see that black, even some of the blue fuzzy, the white fuzzy, usually rots from the inside out. That's degrading the stalks. So here's a field in Iowa. This is the same field, just different parts. It was all vertically tilled. We applied our fungal heavy product. 3 weeks later, you can see on the right where we applied it for where we didn't. So we like to say, and you guys have probably all heard this, you do no-till and cover crops to improve your soil. Well, that's a journey, and it's not always a profitable journey. But what we're finding instead is you put life back in the soil, which then enables you to do no-till and to do cover crops, because the microbes are taking care of the residue, so that when you plant in the spring, the soil is warming evenly and you can plant right through this stuff. I've got a lot of customers that have sold their chisel plow because they don't need to anymore, because the residue is protecting the soil during the winter, but not causing a problem in the spring. Plus, they're getting all the nutrients back in the ground. So, speaking of going back in the ground, microbes really are what glue together the soil. So if we look at compaction, I like this quote that came out of this gentleman from Ohio State. The susceptibility of soils to compaction is the direct result of a biological problem, not your heavy equipment. That contributes it with wet ground, et cetera, you know all that, but it's biological. So what's going on? So imagine you've got 2 5-gallon buckets. In one, we're going to put a really finely ground sand, and then we're going to tamp it down real firm. In the other, we're going to put golf balls, and we're going to try to tamp that down real firm. It doesn't work real well. Now we're going to put water on top of each one. What's going to happen in the sand? It's puddle on top. What's going to happen with the golf balls? It goes right in. That's the soil. All soil is made up of those really fine particles called microaggregates. What we want to do is glue them together into macroaggregates. We want to turn the sand into golf balls. How that happens, it's the secretions from fungi and plant root exudates that are the glue. So fungi secrete the glue that makes the sand into a golf ball. Then the fungi, that white feathery stuff, encases, like the outside coating of a golf ball, these macroaggregates and stabilizes them, as does plant roots. Now the bacteria can't get to them. Bacteria love the glue. It's a great food. What happens when we till? Oh, we break that macro aggregate open. Now the bacteria can get in. They eat the glue. The golf ball collapses into sand. So it's fungi and roots that fight compaction. And we can see this. So the picture on the left is actually pretty close to my house. I live in Northeast Indiana, actually not that far. I live outside of Fort Wayne. This actually happens to be an organic farm that I run by. And I had to stop after Thanksgiving, late November. These are some of the biggest dirt clods I've ever seen. They're huge. And if you look at the face of them, they're sheer, there's no texture. And you could pick these up with both hands, you are not going to crumble them. Think about how much field work is going to have to happen in the spring to plant this field. I'm going to find out because I'm going to be there taking pictures. Why? Even though it's organic, it's tilled. When we till ground, we kill fungi. Fungi live a lot longer than bacteria, but they're wimpy. They don't like to be disturbed. All that white feathery material gets chopped up, and then the bacteria eat the glue, and that's what we're left off with. Compared to the right, where now we've got really good soil. This is a long-term customer of ours. Look at the face where the sun is hitting. All that texture, those are macro aggregates. And you know you can put that clump in your hand and just crumble it. It holds water, air is in it. That's what we want to see. So we did some science on this. So we looked at 44 farms last summer across Iowa. And we looked at bulk density and water infiltration. And we applied the product at planting, which is probably, sometime mid to late April, and we started taking tests. You can see how bulk density over the 7-week period starts to decrease. Look at water infiltration. After about 7 weeks, goes up 64%. In the cornfields, 53% in the beans. Why? Because the fungi are starting to grow during the 7-week period. It's turning the micro aggregates into macro. We're building the golf balls. We're opening up the soil. Healthy soil gives you higher yields. There's all kinds of yield studies on our website. I don't like to go into it because if you take care of the ground, you're going to have a stronger plant that's going to do better for you. This is a satellite image. Now, this is South Dakota, so it's not really Ontario. This is a customer in South Dakota. Is pretty dry, by the way. And this is a guy that started using our product in 2019. Look at the change. This is NDVI. So green is good. It's a healthy plant. Yellow and red are certainly less good. The purple, by the way, is his home. So that's around his house. 2020, he had good moisture, really good year. 21 and 22, severe drought. And look, it's still a huge improvement. Why? Because we have rhizosheets and we have deep roots, and the fungi are working with the plant to take care of the drought stress. 2023, we don't have the map yet, but he actually won the Group 2 soybean yield contest for the state of South Dakota, almost 83 bushels an acre versus county average of about 42. So, by the way, he's also through time cut his nitrogen in half. And because for years he has spread cow manure, he doesn't apply any P or K. Now, he did this slowly, using the biology as a tool to see where it worked the best. Everyone's not going to be able to do this, but you can reduce. How much really depends on your ground. Again, here's something from Ontario. I just got this from last summer. You can see what the grower said in terms of his yield bump, which is pretty good. So I threw a lot at you. What I hope you remember is that microbes really are not snake oil. They are the life in your ground that helps the ground work. And diversity is the key. We need all the bacteria, actinomycetes, fungi, protists. If someone's selling you a powder or a liquid in a tightly sealed jug, it cannot have all of this because they can't survive. This is actually, you think about it, we're under pressure. We got to produce more food. We have to use fewer inputs and be more sustainable. This is taking care of the ground helps you do it while you actually improve the quality of your soil and the value of your farm. And so these are shirts that one of our customers made up, Holganix, so the farm is here for me. And those are his kids, actually down in Tennessee. That's my email. That's our website. At this point, I'd be happy to answer any questions. And thanks for your attention.
Great, Dave. I'll let people put their questions into the chat while I get started. So with Holganix, I guess, Dave, what crops did you start with the product's development, and when you were kind of making your recipes? Why was it needed in those first, I guess, few years when you were just getting started? What was the impetus for it, I guess?
Yeah. So a guy named Barrett Ursick and another guy named Dave Thompson, so I told you earlier, uh, had the largest lawn care company in the mid-Atlantic region of the US. They had over 100 trucks spraying fertilizers and herbicides on lawns around the Chesapeake Bay. And then to protect the waterway, they banned phosphorus. Now, this is something I know that Canadian farmers, as well as a lot of farmers, are worried about in terms of legislators telling us how much fertilizer. He started looking at different things. He found this weird bug juice from an inventor because he wanted to keep his lawn care company competitive, and he saw how well it worked. And it worked so well, he sold his lawn care company and bought the inventor out, who he hired on as one of the scientists, not me, by the way, and called it Holganix. Uh, the company started in lawn care and for years has been used. So if you watch a sporting event in the United States on real grass, they're probably a customer. Major League Baseball, professional football, golf. We've been in agriculture since about 2019. I came on board after I retired from a big ag company and helped us get all the data and stuff. So it was really phosphorus. He had no idea that it was full of phosphorus solubilizing bacteria. He didn't care. He just knew it worked.
And you've given a lot of examples, particularly from the United States. I guess when you are looking at Canada as a whole, I guess what are you seeing in terms of use cases or advantages from maybe getting into a different market that farmers are seeing in Canada? Is your recipe, I guess, hardy enough for that for biological life up here? Is there any differences there?
Yeah, we actually have some really good results in Canada. So we do have a Canadian label, and we sell across Canada, mostly in Ontario. Although we've done some work, in the provinces, the big, prairie provinces like Saskatchewan. I've even sold into British Columbia on cherry trees. So the product works just as well. Really, any plant that needs healthy soil needs biology. And I think that's probably almost every plant. So we see really good results: corn, soy, wheat, some canola, the vegetable crops. Um, I think one of the tricks with no-till is in places where, depending on your geography, if winter comes right after harvest, then you really don't have time to use any microbial product because every microbe needs a little temperature and moisture. So if it's going to snow or the ground is frozen, then you really need to wait till spring. So that's just the only thing that we need to work with people on in terms of when they can get out there and apply it before cold weather hits.
There we go. Dave, thanks again for joining us tonight. We're going to move on to our next speaker, but Dave, if you could put your information in the chat if anyone has a follow-up question or wants to get more information about Holganix, that would be great. And we can keep rolling into our final presentation of the night. And I'd like to invite Boya to share a bit about Ag Leader. Thanks, Dave.